Okay guys, welcome back to my new video of the class 12th first chapter, that is sexual reproduction in flowering plants. I am Hani Modi and I am going to teach you today the very first chapter in the class 12th biology, and I will be also discussing the important question for theory as well as MCQ purpose. So let's get started with this chapter. Okay. As the name suggests, we in this chapter we are going to deal with how the plants reproduce. We all know that reproduction is of two types. It can be sexual as well as asexual. But in mainly this chapter we are going to deal with the sexual reproduction in flowering plants. So there are some key points you need to know about this chapter. So reproduction in flowering plants is predominantly sexual. If we talk about the angiosperm, we all know the classification system of the flowers, plants, animals, everything. We did it in 9th class. So in reproduction in flowering plants is predominantly sexual, and mainly the angiosperm mostly reproduced by sexual reproduction. Chapter we are going to deal with the gamete formation, pollination, fertilization, seed and fruit formation. If I need to tell you the important questions or important topic among this, gamete formation, pollination and fertilization is the most important. You need, and it's compulsory for you to remember it because it's gonna come in your either MCQ or your theory paper. And I do suggest that if from fertilization, double fertilization is the hot favorite question. And from pollination, yes, types of pollination. If they want to ask in theory exam, they are gonna ask you its type and it described by giving examples. And if they are going to ask you in MCU, they will just give you example and tell you which type of pollination it is, or what are the factors, or who are the media through which this type of pollination occur. We will see in this all this in the pollination video, which will be coming in next video. You will see it. And uh, why the sexual reproduction is important, it mainly ensures genetic continuity, variation. Moving forward, significance of sexual reproduction, it promotes genetic diversity. We all know its evolution and adaptability. We all as the environment changes, we humans also adapt ourselves to stay in this environment. It's so same thing goes for plant. Plants also needs to adapt themselves to stay in such extreme conditions. They're like sometimes we get to too high temperature, and sometimes we get too low temperature. They need to adapt themselves. Also, you need evolution for this adaptability. Next, important for crop improvement and hybridization. Hybridization is another great topic which will be covered in pollination topic. You will see in that. So till then, stay and continue this video till you get there. Okay. Next is flower, the reproductive unit. Okay. Mainly, let me get you here only diagram. Okay, flower has a petal, we all know. It has a sepal, we all know. Now, it has a male reproductive organ. Great. Now, what are the male reproductive organ? Male reproductive organ is known as stamen. What it is known as? Stem. Now, stamen has two parts. Which are they? Anther as well as filament. Got it? Now let's spot it in diagram. This one is anther, yes, you can see. And this, the lining coming from the anther is the filament. Done with the male reproductive organ of the flower? Cool. Now coming towards the female. The female reproductive organ has a three parts. Stigma, style, and ovary. As the most famous term we use in the reproductive unit of any plants or animals, male reproductive organ is known as androecium. Female reproductive organ is known as gynoecium. Great. Good. This is stigma. I told you in the top to bottom is anatomical position, stigma, style, This is style. And exposed part is stigma. Then comes the style, the stick, which connects the stigma and the ovary. And the bottomest part is this ovary. Good. Okay. Now, the any plant, basically, let's talk about term as it has a flower. Any flower can be unisexual or bisexual. What does it mean? Bisexual means it contains both androecium plus gynoecium. Unisexual means it contains either androecium or gynoecium. Okay, let's, after seeing this figure, can you tell me that whether it is a unisexual or bisexual? Of course, it's unisexual, but because it contains both androecium and gynoecium. Okay. Moving forward, the for very first topic we are going to see is pre-fertilization events. So basically, what is pre-fertilization events? These are the events we need before doing the actual transfer of gametes. Cool. It involves gametogenesis and gamete transfer. Gametogenesis means formation of male and female gametes. We earlier saw the structure of the female and the male reproductive organ, but we need to know that from these organs how the gametes are going to get produced. We know that the gamete is the most important cell structure which we need for fertilization. So, formation of male and female gametes is called gametogenesis. Gamete transfer means movement of male gametes to the female reproductive organ. We all know that mostly male gametes move towards the female reproductive organ. This is the basic knowledge you must know, and or if you don't know, we will see further. It's also going to come in one of the tops. Okay. Till then, just remember that the male gamete always go travel to the female reproductive organ for fertilization. In either flower or animal, it is the commonest thing. Okay. Coming towards the male reproductive organ, also known as androecium or stamen. Okay. Yes, this is the perfect picture which is easy for you to understand. As I told, this is the anther, this is the filament, or you can also say the stalk. Now, each anther, if we cut this anther like this in transverse manner, then we are gonna see that it is a bilobe. What does it mean? That you can see one lobe, two lobe. So it is a bilobe structure. Now, this lobe also contains this two theca. We can say that two theca. Each lobe contains two theca. First theca, second theca. And that's why it is also known as dithecus. So tell me overall how many lobes are there? Four lobes are there. Good. There are two lobes basically, and there is divided into two lobes, and each lobe is further divided into two theca. So there are overall four lobes we can say. So each lobe has two theca, that is also known as dithecus. Now moving towards, let's see what's written in the slide. It is composed of filament and anther. I told you. Anther is bilobe. Discussed each lobe with two pollen sacs. Now this theca, what it is also known as pollen sac because it is the place from which all the male gametes are going to be get formed and released from here. Okay. Now it contains microspore mother cell. Okay, this is the very interesting thing you need to know. Which let me get you the diagram first. Okay, before moving further, let me just tell you properly each and everything. Okay, the anther is a four-sided tetragonal structure consisting of four microsporangia located at the corners of each lobe. Okay, we first, second, three, four. Total there are four theta we can say, but we are going to say it as a microsporangia. Okay. Now this microsporangia will be located in the corners of each lobe. Okay. Yes, this diagram is cool. Now, okay, this is the transfer section. Let's just say study this part only. Don't see this all things. It will be give you somewhat trauma, but currently you just need to focus. Okay. Or better, I can say this one. Okay. Okay. Transfer section of anther. Now this is one lobe. Here there will be microsporangia located at the corners. From here, pollen grain will be made and released. Uh, okay, this that is a formation of this pollen sac, which goes on as the further the anther gets matured. Okay, now let's deal with this structure of this microsporangia. As I told you, that microsporangia is this, and this. So basically, how does the transverse section of anther look like? First of all, the outermost layer is epidermis. Okay, here comes the next important question. Four layers of microsporangia. And it's the important one. It can be asked in the short questions, or it can be marked the odd one out, which one is for one of the layer of the microsporangia or not. So you must know it. The outermost layer is epidermis, followed by endothelium, followed by middle layer, followed by tapetum. Tapetum is the innermost layer of the microsporangium. You need to know this is the most. And the question will be asked, which are the layer which do the protection? So you will answer epidermis, endothelium, and middle layer do the protection. Tapetum doesn't to give the protection. The tapetum is the area where it nourishes the basically the pollen grain. It contains the nourishment content in it. Okay. Which are the layers which do the protection? That is epidermis, endothelium, and middle layer. Done. Inside epidermis, there is a sporangious tissue. We all know that. What does this actually do? This further do mitotic division. So yeah, we were here. I was telling you about this pollen grain and this sporangia. Now, what does actually happen? Okay, let's see. Mm, 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 mm. Mhm. Now we deal with the basic structure of how this stamel looks like. Now we are got further going to see the microsporogenesis, which means formation of microspores or male gametes from microspore mother cell via meiosis. Now, we know, I told you guys that it contains the sporogenous tissue. Now, this sporogenous tissue, also known is cell of this is known as microspore mother cell. What it is called? The cell of the sporogenous tissue, it's called microspore mother cell. Now, what it do? It do the meiosis. We all know what meiosis is, and it lead to formation of four tetrad cell. Four tetrad cell, or I can say tetrad of four haploid microspores. Now, this microspore got converted into pollen grains. Each pollen grain has two grains, that is vegetative cell and generative cell, which we will discuss later on. But first, let me focus you guys in this thing. Let's revise. Microsporogenesis is formation of microspore from microspore mother cell via meiosis. Now, what is microspore mother cell? Microspore mother cell is nothing but the cells of sporogenous tissue. Getting my point? This microspore mother cell do the meiosis through which it get converted into tetrad of four haploid microspores. Now this microspores as the एंथर मैच्योर जब ये एंथर मैच्योर होगा ये तो जब यंग होगा तब वो करेगा जब वो मैच्योर हो जाएगा तब ये माइक्रोस्पोर प्रोड्यूस करेगा पोलन ग्रेन और ये पोलन ग्रेन रिलीज होंगे You can see the releasing of the pollen grains. Yes, that's what it is. Now let's revise. Outermost epidermis, followed by endothelium, next middle layer, and then the tapetum. Protection is done by these first three layers, and the tapetum, the role of tapetum is to provide the nourishment. Cool. Now the how the microsporangias happen. Okay, microsporangias by formation of microspore from microspore mother cells. What is microspore mother cell? It is the cell of sporogenous tissue. What is sporogenous tissue? This is the tissue found in the pollen sac of the anther. Then we all saw that. Now MMC will do the meiosis and produce tetrad of four haploid microscope. This microscope, when the anther matures, will be converted into pollen grains. Now, this pollen grain will be released when it's a appropriate environmental condition. Now, what is the basic structure of pollen grain? It contains the outermost exine layer, which is a tough layer, and it is made up of sporopollen. This is the important question or MCQ which can come. And if you have the vivas in your school, that of course they are going to ask if the chapter is then in the exam. Exine is the outermost layer. It is produced from sporopollen. Now, what is the spore function of sporopollen? Sporopollen basically protect and इस यही रीजन है कि पोलन ग्रेन्स बहुत ज्यादा टाइम तक रह सकते हैं with adverse environmental condition. Now, there are some pores here, basically germ spore, where sporopollen is exempt. So, germ pores where on the exine where sporopollen is absent. That this is the place where sporopollen is absent. Now the inner layer is called as intine. Intine is made up of cellulose and pectin. Okay? And it is a continuous layer. Okay? Now, as we saw, let's discuss about the structure of pollen grain. So this is exine. Sorry, I marked it wrong. Exine and intine. Now, the thing is, as the pollen grain matures, it goes into changes, cellular changes, for which the each pollen grain will have a two type of cell. It will be, first of all, it will be vegetative cell, next generative cell. Now, generative cell will divide into two male gametes. Okay, pollen grain, vegetative cell, generative cell. G for generative. Generate means मतलब generate करेगा. क्या? दो male gametes. Two male gametes. Okay. Now, what are this through vegetative and generative cell? Let's see and clear from the diagram. As the pollen grain matures themselves and or we can say as go into cyclic changes or cellular changes, the two cells develop. First, vegetative cell. It's the bigger one and it has a large irregular shaped nucleus as you can see and it contains the abundant food material. अभी खाना इसके पास है, food है, irregular nucleus है, तो अभी जनरेट तो करना पड़ेगा ना, तो ये जनरेटिव सेल जनरेट करेगा दो मेल गैमेट्स को. Okay, generative cell will generate two male gametes and all the nutrition will be provided by this vegetative cell. Okay, now as we saw in the picture, generative cell is small and floats in the cytoplasm of the nucleus and it is spindle shape. Say like basically you can see spindle shape here. Okay, for other muscular cells shaped like. If you have studied 11th class properly, you might remember. It is also spindle shape. Like this. Oh, sorry. like this. It is also spindle shape. So remember like that. Okay. So this is the what the actual microsporogenesis looks like. Mm. Okay, so let's revise structure of pollen grain. Important part: exine outer layer made up of sporopollen. And where the sporopollen is where it is absent, it's called germ pores. Next layer, innermost layer, engine made up of cellulose and pectin. Another thing, exine is for protective function, and it is made in such a way that it can live in adverse condition. This is the electron microscope of few pollen grains. See, we इस एक डायग्राम ये है, but इलेक्ट्रॉन माइक्रोस्कोप में ऐसा दिखता है. Okay, you can see this for exine and mode will be engine. Okay, another thing I forgot to mention to you guys is that this in 60% of flowers जब भी ये टू सेल स्टेज होता है, टू सेल स्टेज मतलब जब वेजिटेटिव सेल और जनरेटिव सेल ही होते हैं तभी ये रिलीज हो जाता है. और बाकी के 40% ऑफ फ्लावर्स में ये थ्री सेल स्टेज होता है. थ्री सेल मतलब क्या? वेजिटेटिव सेल प्लस टू मेल गैमेट्स. ये ऐसे रिलीज होता है. मतलब कि अगर ये टू सेल में रिलीज हुआ है तो वो फीमेल रिप्रोडक्टिव ऑर्गन में जाके जनरेटिव सेल टू मेल गैमेट्स में कन्वर्ट होगा. और अगर वो थ्री सेल स्टेज में कन्वर्ट हुआ है तो डायरेक्ट मेल गैमेट अटैक करेगा फीमेल रिप्रोडक्टिव ऑर्गन को. डन? समझ में आया? सिक्स ये यू नीड टू रिमेम्बर दिस क्योंकि आगे जब फर्टिलाइजेशन आएगा तब ये पॉइंट इंपॉर्टेंट हो जाएगा कि यूजुअली सबसे मोस्टली 60% में से वेजिटेटिव और जनरेटिव सेल में ही पोलन ग्रेन पहुंचा होता है और तभी रिलीज हो जाता है. और वो रिलीज हो जाता है उसके बाद फीमेल रिप्रोडक्टिव ऑर्गन में जाता है. फिर वो जनरेटिव सेल गेट्स कन्वर्टेड इनटू टू मेल gametes. But in few of them, it's already divided into two male gametes and then released. Okay. Now coming back to the pollen viability and storage. First of all, see pollen grains है ना, ये wheat. For example, let's take example of wheat and rice. Wheat और rice जो है ये दोनों में इनकी सिर्फ अगर ये रिलीज हो गए, रिलीज होने के बाद 30 मिनट्स तक वायबल है. एंड इन केस ऑफ रोसेसी and leguminaceae, refer textbook for correct spelling. Precautions to be taken. ये months तक वायबल है. डिपेंडिंग ऑन द प्लांट्स उनकी वायबिलिटी. वायबिलिटी मतलब क्या है कि वो एक बार रिलीज हो गए एंथर में से तो वो कब तक रिप्रोडक्टिव ऑर्गन में पहुंचने से पहले खुले एनवायरमेंट में झूम सकते हैं. ओके, प्रॉपर वर्ड रह सकते हैं. ओके, व्हीट और राइस लाइक सीरियल्स पे 30 मिनट्स तक रह सकते हैं. और रोजेसी और लेग्यूमिनेसी वाले प्लांट्स में मंथ्स तक वायबल रह सकते हैं विदाउट गोइंग टू द फीमेल रिप्रोडक्टिव ऑर्गन. ओके, नेक्स्ट इज उनका प्रिजर्वेशन. मेनली एनवायरमेंटल कंडीशन पे मोस्टली डिपेंड रहता है. लेकिन कुछ बहुत सारे प्लांट्स को लिक्विड नाइट्रोजन में स्टोर किया जाता है इन -196 डिग्री सेल्सियस और इसे कहते हैं क्रायो प्रिजर्वेशन. ओके, इसे क्या बोलते हैं? क्रायो प्रिजर्वेशन. और ये यूजफुल होता है हाइब्रिडाइजेशन में. जब भी कोई भी एक्सपेरिमेंटल स्टडीज या रिसर्च स्टडी करनी हो या फिर हमें प्रॉपर क्रॉप अच्छा क्वालिटी का ब्रीड करवाना हो इन दैट कंडीशंस. और अगर हमें पता हो कि फ्यूचर में इसके सीड्स वायबल नहीं होंगे, ये इसके प्लांट का fertilization नहीं होगा, in that case we do the cryopreservation in liquid nitrogen in -126 degrees Celsius and it's useful in hybridization. So this is all about the male gamete. Let's just move to the slide. First of all, structure of this. Now, pre-fertilization, that is gametogenesis, gamete frontal, what are structures of stamen, filament anther, bilobed होता है, diathecous होता है, each pollen sac में स्पोरेजिनस टिश्यू होते हैं, ये रहे स्पोरेजिनस टिश्यू, ये स्पोरेजिनस टिश्यू एमएमसी बनाते हैं, एमएमसी मियोसिस करते, टेट्राड नंबर ऑफ हेप्लॉइड सेल बनाता है, ये पोलन बनाता है, एनवायरमेंटल कंडीशन में रिलीज हो जाता है। पोलन कंटेन टू लेयर एक्साइन इंटाइन, इसमें स्पोरोपोलिनिन होता है, इसमें स्पोरोपोलिनिन होता है, इसमें सेलुलोसापेक्टिन होता है, जर्म फूड प्रेजेंट होते हैं एक्साइन में, जहां पर स्पोरोपोलिनिन एब्सेंट होता है। ओके, स्पोरोपोलिनिन प्रोटेक्टिव फंक्शन करता है। नेक्स्ट, ईच पोलन ग्रेन में दो सेल होते हैं, वेजिटेटिव सेल, जनरेटिव सेल। जनरेटिव सेल में दो मेल गैमेट्स में डिवाइड होता है, 60% टू सेल स्टेज में रिलीज होता है, 40% ऑफ पोलन ग्रेन रिलीज इन थ्री सेल स्टेज। Okay. Electron micrograph of few pollen grains, and this is how the pollen grains mature itself and form the vegetative and generative cell. Vegetative cell contains the nutritious food, irregular shaped nucleus. Generative cell generates into two male gametes. Now, pollen viability, wheat rice 30 minutes तक viable है, pollen grains of wheat rice 30 मिनट तक वायबल है, और रोजेसी और लेग्यूमिनेसी प्लांट्स में months तक वायबल होते हैं। Then cryopreservation is done in liquid nitrogen and useful in hybridization. Now, there are some of the pollen products found like pollen tablets, which improves the performance of athletes and horses, mainly race horses. इन दोनों के ये परफॉर्मेंस को इम्प्रूव करते हैं। ये जनरली इंडिया में नहीं पाए जाते हैं कि, लेकिन कभी कभी फॉरेन मार्केट्स में अवेलेबल होता है। टैबलेट एज वेल एज लाइक दिस सीरम इज ऑल्सो अवेलेबल। ओके, okay, so this was all about the male parts of the pre-fertilization event. In next video, we will see the female reproductive system as well as generation of female gametes. So next video will be about female reproductive organ and its gametogenesis, and then next to it will be pollination, and then we will see the fertilization and post-fertilization event in flowering plants. So till then, do read it from the textbook, revise it, solve the MCQ as much as you can, and do comment if you have any queries about it, and do like it if you really enjoy it. Till then, bye bye.